Hello everyone, and welcome to our first seminar in the DNA Dialogue series for 2023. In the spirit of reconciliation, I acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. I'm joining you from the lands of the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present and extend that respect to all First Nations peoples joining us today. So DNA Dialogue is a forum to share information and insight into developments in health genomics from about the world. These seminars are held live via Zoom on the last Thursday of each month. And while previously we held these seminars early in the morning, for this year we're trialling sort of different times to accommodate not only our guests and attendees from around Australia and around the world, but also to attract speakers from different parts of the world. If you're a tweeter, please use the hashtag DNA Dialogue and tag us at AusGenomics, sorry. Do please ask questions anytime during the talk using the Q&A function, and I'll get to as many of those as I can during question time. So now, without further ado, it's my very great pleasure to invite Professor Aarno Palotie to join me on the screen. Aarno is the Research Director of the Human Genomics Program at the Institute for Molecular Medicine in Finland. He is also a faculty member at Mass General in Boston and an associate member of the Broad. Hello, Aarno. Aarno has a long track record in human disease genetics. His research leverages the unique Finnish population and healthcare to improve our understanding of the genetic component underlying common diseases. He's chaired several large international research consortia and is the scientific director of the FinnGen project, which we'll be hearing about today, a large public-private partnership aiming to collect and analyse genome and health data from 500,000 Finnish biobank participants. The study has already identified hundreds of disease-associated loci where the associated variant is enriched in the Finnish population, including a large amount of damaging coding variants. These provide potential for further functional studies and new insights in disease mechanisms. So, Aarno, thank you so much for joining us early in your morning. We're super excited to hear your talk. So I'll throw to you now and I'll see you again at question time.
Thank you, Tiffany. And good afternoon, everyone. It's lovely to be invited to this forum, and it's always great to talk about a subject which is close to your heart and that you are working with. And also I have close connections with many scientists in Australia and feel very warm about those collaborations. And today, as Tiffany was explaining, I would tell you about the FinnGen project, which is a large nationwide biobank study. But maybe first driving into you to kind of remind what Finland is about. So Finland is a small country, 5.4 million. However, the surface area is relatively large on the size roughly of Germany. And that means that we have certain interesting aspects when we try to do something nationwide. One is that the very uneven population density in the country, meaning that most of the people are living in the southern area and on the coastlines, whereas, for instance, Lapland is clearly something which is less inhabited, and for instance, how to organize the healthcare in an even way. We face actually some of those similar challenges you have in Australia, just in a much larger surface area. And then the question is that why Finland for genetics studies? So basically, if we look at things where we tick the boxes, that we have the biobank system, which I'm going into in more detail later. We have a universal healthcare system. And very importantly, we have a unique personal identity code, which is very typical for the Nordic countries, where every individual has their own code, quote unquote, social security number that is very widely used. It has a special population structure. I'm going into that a little later, and the biobank system is enabling recall. Whether that's made easy, that's another story, but basically the recall is there. So these are the kind of fundamentals why Finland is maybe larger in this field than its actually the size otherwise would predict. Let's first dive into the genetic isolate aspects. So as said, Finland is located in the northeastern corner of Europe, and in a way has a peninsula aspect, and it's surrounded by waters, and that has kept Finland rather isolated for centuries. The main founder population has come from south, from Europe, but also there is influx from east. And what happens obviously in a genetic isolate is that there are certain variants enriched in that type of a situation. There's another additional element to it. In the 16th century, when Finland was still a part of Sweden, the eastern border of Sweden, the Swedish king demanded Finns to settle, to move in Finland to protect the eastern border from Russia. And that resulted in additional multiple bottleneck situations where people from south were moving to east and north, and that we still see today. And importantly, in all population isolates. So there was a rapid expansion of the population starting from the 18th century in a very similar way than in other Nordic countries. Yet the other Nordic countries don't have this isolate feature. And what it ends up with is that there are specific damaging genetic variations that become enriched. And they are also easier to discover, kind of the background noise is lower. But it also enables the genetic quote-unquote imputation much easier. That means that reconstructing kind of the genotype from a GWAS array is much more efficient in an isolated population like this, where the haplotype structure is much larger. And that's why we haven't had to sequence everyone. Just sequence a population panel currently of some 8,000 individuals who have deep whole genome sequencing, against which we impute the GWAS genotype. The other element comes from the healthcare system, which was reorganized in the late 1960s, and it resulted in something which we call national registers. And this is quite unique for the Nordic countries, where every individual, wherever she or he purchases, for instance, a prescription drug, wherever in the country the person does it, the purchase goes into a national register, which has stores it in a digital form, typically as an ATC code and core code, and in the case of hospital visits, in an ICD code. So the same thing is whether the person stays at the hospital, visits a specialty clinic, and lately even the primary care units. And these registers have been initiated, the earliest from the 1950s, the cancer register, then hospital discharge registers from 1960 and so forth. And that's displayed here with the arrows over here. What that means is that we have healthcare information, healthcare usage information from every single resident in Finland from birth to death. And that obviously provides an interesting opportunity to also for research, even if these registers were originally developed and founded for administrative purposes. And then even if the registers are in silos per se, the individual identification number, which we all Finns have, then is the glue or the key from register to register. So there are kind of four cornerstones that we stand on: the population isolate, the health registers, which also has then stimulated the long tradition of epidemiological studies, which then have further developed into biobanks. And then, of course, there are decades of genetic research in Finland. So the population is also quite used to the fact that we have geneticists who work on our genomes. But having all this, if we wouldn't have a government backing of that, it's that which kind of thinks and have had the basic idea that healthcare might not only be a cost and a burden for the society, but also through a thinking that good healthcare, this type of healthcare information also provide us opportunities, meaning that opportunities for research and developing healthcare in the long run, and also potentially from an economical perspective. So what happened a few governments ago was that 3 ministries together, which, I mean, people who work in ministries around the world know that when 3 ministries together do something, that's already unusual. But this is what happened. They together started a plan that how could we develop the healthcare sector strategy to something that would be of benefit for the society and economy. And the government backing then resulted in a series of bills. First, the National Genome Strategy was kind of laid out in 2014. Then the Biobank Act, which was important and something we very much are basing our activity on, and that then secondary usage of this health register data. Primary usage is obviously for the immediate patient practice, but then secondary uses it would be for research. And then there was a bill of National Genome Center that hasn't really been flying forward just as we thought, and maybe good so. And the Finnish Biobank Act is in some ways unique. And the key is that there is a wide consent that where the participant, which the participant gives when she or he gives the sample and consent the data to be used for medical research in a broad sense. Now, those ones who know GDPR so might wonder why that's possible, but that is possible. We might return to that if that is of interest. Anyway, this is a key thing here. And on the same time, the person consents that there is a possibility to recontact for further studies, and also that the possibility to collect samples and data from the healthcare. And even the law says that it's okay to collaborate with industry. And this has then resulted in a network of biobanks. We have some 10 or so biobanks around the country. and they are then coordinated with something which we call the FinnBB, the Finnish biobank cooperative. So access to the digital health data based on the strategies laid out by the three ministries initiative of growth, health sector growth strategy. So a data permit authority was established. It's called FinnData. Basically, they then provide or you apply for access to this register data from FinnData, and then they approve it or disapprove it. This was started a few years ago, and when FinnGen started, this wasn't still in existence, but the idea is that this would be one portal so that we don't have to go to all different register holders to ask for their permit, but FinnData takes care of that. There have been ups and downs with the system, and because it's relatively new, so it was a little bit understaffed. Things are getting much better, but we also have learned that the legislation would need some fixing. So we can maybe skip this one. The idea has then been that there would be a safe kind of data, trusted data environment where the data from these registers would be used. That does exist in the Center for Scientific Computing in Finland. The reality is that it's not quite yet developed to a stage where, for instance, FinnGen could use it. So FinnGen has its own trusted environment. And but those ones who work in ministries know that even if there's a goodwill, so there are things to overcome, because some of the activities and some of the registers are managed by different ministries. Ministry of Health and Welfare has one domain. Ministry of Education has one domain. For instance, having the computing environment being in there under their governance, and the Ministry of Employment and Economy has the most important funder for Finnish kind of innovative research, which has a business angle. Academy of Finland being then funder for the pure basic research. So in other words, in addition to the four cornerstones that we have, there has been additionally legislative improvement that has made it possible to do a study like FinnGen. The reality, though, is that everything is not always so glamorous as it maybe is in these presentations. There's quite a lot of work that in the background that goes that we fulfill the GDPR, the European legislative environment, and the national environment. And one of the challenges in Europe has been that each nation then has, on top of the overall GDPR, national regulations, which don't always match each other. So when you want to do cross-national things, so things are not totally easy. And that's why the European health data space is an initiative that the Commission is working on. What that will be, when that will be, we'll see. But there is a strong initiative, and partially the Finnish model is one of the models that has been discussed for its function. Okay, then we finally dive into the FinnGen project, which really, as Tiffany said, 500,000 individuals, which is roughly 10% of the Finnish population, having their DNA assayed on an Finnish developed GWAS array using, as said, the population-specific deep sequence imputation panel to get up to some 20 million genome variants, and using primarily the National Health Register data as a phenotype resource. And this provides then the study sample of 500,000 individuals for the association analysis. This is a public-private research project, and all these words are quite important here. First of all, it's that both the public and the private invest their time and energy and intellectual capacity in this. It's a research project which has been key for the permits because this register data secondary use can be used for research, and actually nowadays also for innovation. That means that all Finnish biobanks, all university hospitals, all and the Institute of Health and Welfare, which is under Ministry of Health and Welfare, are partners in the project, and the University of Helsinki is coordinating it. There are currently 13 pharma partners as full partners in the project. We started with 7, but 5 more have joined, and it's a true joint project in the sense that we together plan what will be done, how it will be done, what are the research questions we like to tackle. And Business Finland has been from the Finnish part kind of quote-unquote governmental funding with roughly 20% of the whole project so far. The project was initiated in 2017. The permits are good for 10 years so far, so that our permits extend now to 2027, and what happens after this is now initiated as a discussion how that goes. The first 6 years have been to construct the infrastructure and collect the samples, and also develop the analysis and perform the first analysis. Now we are in August 2023, we hope to move into the FinnGen 3 phase, which will not have much of data collection anymore. It's a main analysis phase and include also functional analysis. In other words, using the resource that we have built to answer scientific questions. And so not expanding it, but going into depth. And again, we went through the National Health Register data. This is the fundament for the phenotype data in the project. And where are we with the sample collection, which was initiated in 2017? We are relying for some 200,000 of what we call legacy collections. Those were mostly in the Institute of Health and Welfare. They were population cohorts or disease-specific cohorts, but in their biobank. Then there are prospective collections, 250,000 of them from hospitals and 50,000 from blood donors. And we currently, the biobanks have more than 550,000 samples already. So basically, next week the collection will end, and that will be the end of the collection, so that we all of the rest will then go into genotyping and so forth. One characteristics when working in a public-private partnership is that our timetable is tight, and we need to follow it. And one of the characteristics has been that there is a new data set and data freeze, so to say, every 6 months. We have been able, and this has been key for the success, to be typically a little bit ahead of the curve, always hitting the timeline and keeping the schedule. And that of course is key when you plan that the project could be even taken further. The current dataset, or actually the dataset which is still the one we mostly use, the new one came just a week ago, but I don't have the numbers for that yet quite. So was some 430,000 phenotype and genotype data in the data freeze, with more than 4,000 disease endpoints constructed from these register data and over 20 million variants. And they are always then meta-analyzed with the UK biobank and Estonian biobank data. So we have actually 2 more data freezes to go then, and then we are in the 500,000 data freeze. So we think that this provides an infrastructure for research, and how to do that different, for instance, from UK biobank. So one of the key things is that there is a secure data analysis environment called which we call the Sandbox, where the individual level data lies. That's in the red box here on the right. So every researcher, when approved from all participating partner institutions, can access the individual level data, can analyze it. There's a number of tools there available, but cannot download the data, only results. Individual level data stay in there. And at the time when we started, so the Google Cloud provided the most secure environment and the best kind of opportunities for security. Then there is what we call a green cloud here in the middle, which doesn't have any individual level data, but has all the summary statistics and results. That is something that I, for instance, browse on a daily basis. I rarely go to the individual level data part. Of course, our analysts do it, but this is the typical view for someone like me to look at the results. And so each partner has their own kind of sandbox area where they have both the green, the summary level data, and the individual level data that they can use for their analysis. So in a way, we can brag that the FinnGen Sandbox has solved some of the problems that some of our EU neighbors have claimed unsolvable. So we, for instance, have 150 users outside EU that can access the data, because one of the regulations in EU is typically that individual level data is easier if you access them within the community, EU community, than from outside, from other areas. And it does fulfill all national laws and regulations and the EU by GDPR. And the same system is actually already adopted by East London Genes and Health as their trusted research environment. And now non-individual summary statistics has already been loaded by over 5,000 non-FinnGen scientists from all over the world. So further, yes, it's good to have an environment, but you need tools to analyze and browse the results. And there has been a huge investment of time and money in developing these tools that results are browsable, and for analysis, the individual level data is analyzable in a way that many scientists can do. And currently, there are some 500 scientists who work with the red level data and doing their custom analysis. So if you compare the UK Biobank and FinnGen, so both are roughly the same size. The FinnGen is more skewed towards older and enriched for diseases. That was deliberate, with the idea that some 300,000 or so, 250,000 were recruited from hospitals and clinics. It's also a bottleneck population, not like the British population is primarily a European population. Primary, there is of course more color to it. And there's a longitudinal detailed data from almost, well, 1950s, 1960s, plus some clinical lab value data for specific areas. This has been GWASed. The UK Biobank has been both GWASed and exome sequenced. We haven't done the exome sequencing yet. And maybe it's also of not quite the same benefit as in a more mixed population, as we can already identify rare low-frequency variants with our strategy. And there's unfortunately no possibility to download individual level data. That comes from the regulatory environment, but the Sandbox provides an environment for individual level analysis. So let's look at a little bit on the results. So it's clearly harvesting time with 430,000 Finns included and 50 years of register data. So you expect that you start to find things. And a halfway point was actually just highlighted on the 18th of January in a series of Nature group papers, 2 in Nature and the rest in other Nature family journals. If people are interested, so can dive into to those papers to have a look on more details of how FinnGen is constructed. This was done with data freeze 5. We are now in data freeze actually 11 already. So that was, so to say, halfway report. And as said, a wealth of new discoveries with these 430 participants, more than 400 Finnish specific gene associations and so forth. But let's dive a little bit into what the isolate provides for, say. So some of the novel findings include diseases like IBD, pulmonary fibrosis, glaucoma, and heart disease, and there are both protective and risk variants. So one of the first ones that we encountered was in inflammatory bowel disease. There was this TNRC18 signal, which we first thought that is an artifact because it was so strong. But whatever we did, it turned out that, oh, it's not an artifact. And it's the most significant IBD risk factor in our country. 7% of cases carry it. And why wasn't it identified earlier? Well, simply because it's so strongly enriched in Finland. It's also detectable in Estonia, but much less in other parts of Europe. And the frequency in other populations is simply so low that there was not enough power to identify it, whereas here it was an obvious hit to find. So maybe looking at a couple of more examples from variants enriched in Finland that where potentially new genes and new variants and new biology can be found. First of all, thinking on overall that most of the variants are in the roughly same frequency as in other parts of Europe, but then there is a tail on the right here where there is an enrichment of variants that are low frequency, and especially importantly, many of them are coding variants, which means that there hasn't been enough time in the population to still to kind of dilute them out. And of all associations, so about 12% of the significant association contain a coding variant in a credible set. Of course, that doesn't mean that always the coding variant is the causal variant, because it's in the credible set. That needs more work. And there are some over 500 unique coding variants, over 500 genes, and typically their allele frequency is less than 5%, and 2/3 of them are enriched in Finland by more than 2 times compared to other European populations. So, of course, protective loss of function variants are of special interest, human quote-unquote knockouts, where no functional copies of the genes are. And so it's not a terribly rare event. There are quite a number of them. And what is especially interesting, that if no adverse effect of health has been identified, so they might be interesting for drug targeting. The poster boy of this has been PSCK9, and of course many of drug companies have wished that there would be more of this type of example. The progress have been slower, but indeed there are variants also in Finland that we have identified around this. And one of the first ones was a protection for glaucoma, which then in the ANGPTL7 gene, which also lowers the intraocular pressure, and there is no other or no negative consequence that we can see in the data that this would predispose to any other diseases. But then looking on a different example, and what biobank studies provide an opportunity is the pleiotropy, studying the pleiotropy. And this is something that we in the beginning had quite a hard time to explain to authorities, especially the ombudsman type of authorities, that we are not just focusing on diabetes, or we are not just focusing on heart disease. We are disease agnostic. We are focusing on everything. And one of the key things, of course, is then that you have the chance to look at pleiotropy, which you would obviously not if you would be focused only on one disease entity, where the same variant has different effects in different diseases. There's a Finnish enriched locus for idiopathic pulmonary fibrosis, the SPDL1 gene. And interestingly, this is a risk variant for IPF, a clear risk variant, but it's protective for all cancers. And that obviously provides an interesting biological aspect to look at what is something that promotes fibrosis in the lung but protects for all cancers. And this is exactly what can be studied from a pleiotropic perspective. And this last figure showing kind of a FIWAS type of graph from the FinnGen browser. These are, by the way, many of these are now publicly available data freezes, so any of you can go and browse them. The longitudinal analysis is another aspect that we have. I was already explaining earlier, and we are pretty much in the beginning of using these aspects, but some examples do exist, and especially when thinking of that there's certain genetics which predisposes to disease susceptibility. But is it the same thing, same variants that contributes to disease progression? It's probably so that in some diseases, the same susceptibility genes that drive towards diagnosis also contribute to progression, but there are several where this is not at all that clear. And the longitudinal aspects are one of those things that we can study from this perspective. A very early example is identification of variants that contribute to disease progressions in arthrosis for those ones where the hip replacement surgery occurred earlier. And there are more examples, but we have, as said, more in the beginning of this type of analysis. It also has needed some methodological development. Another question that we have been able to look at in large data sets is that if we talk about especially in a isolated population, the typical thing is that we have an enrichment of inherited diseases which are recessive. And then the question is that is it so black and white that you need 2 copies for the disease in recessive in a disease which is called recessive? And here specifically Henrika Heine looked at variants that are in the rare fraction of or less frequent fraction, low allele frequency fraction in the Finnish population. And just looking at a few examples where some of these are known. So the question is that looking or one of the observation is that, for instance, 5 known recessive disease variants have genome-wide GWAS hits also. These are SERPINA1, 3 of these 5 have effects also in a heterozygote state. There are more of them, but these are just 3 examples. SERPINA1 is actually well known for cholelithiasis and emphysema. The XPA, which is typically seronera pigmentosum gene for skin cancer in homozygote form, but also increases risk for skin cancer in heterozygote state. And NPHS, the Finnish nephrotic syndrome, is the major hit also in adult initiating nephrotic syndrome in a heterozygote state. And this is just one example of the xeroderma pigmentosum gene, where in heterozygote state, the skin cancer risk is elevated compared to the non-carriers. So these are the type of things, again, that you can study in a longitudinal setting, in a large population study in an isolate like this. So we think that this has developed a centralized research resource now that can be used by all Finnish researchers. And we, of course, work towards clinical application and risk assessment, and there are already quite a lot of PRS type of studies done that guide us towards thinking that for instance, cancer screenings would there be an opportunity to look into changing the screening regimes that we currently have based on some background risk estimation. So what is different in FinnGen? First of all, it's nationwide, it's a special population, enrichment of disease cases due to the recruitment scheme. It's a public-private partnership with a really academic atmosphere, and these are when the pharma has told us what they are especially pleased with is, of course, a couple of things. One, that we have been on time. The other one is that it has been an academic atmosphere where the colleagues from different pharma can come together around the same table and brainstorm scientific questions, and all partners are working together here, and that has been one of the things. And then the strong core analysis group. So different from UK Biobank, we provide ready analysis, which especially for the pharma has been really important, so that they don't have to redo the analysis, they can just browse most of them. So now we are working, as said, towards the next phase, the next 4 years, hopefully that will materialize, where really the questions are to characterize all the rare coding variants, to study their function and pleiotropy, and learn genetics of disease progression. And then, of course, reminding that Finland, yes, I admit, even I admit that there are some successes in FinnGen, but it's definitely not enough. And like everyone who is running these type of biobank studies knows that typically no biobank has a sufficient number of cases for those diseases where there is unmet medical need, and we would need more data and information. And that's one of the initiatives is the global meta-analysis initiative, where also we have Australian members in this, and we hope that this type of studies with further help towards new discoveries and bringing the field forward. And then I'd like to thank you for your attention and happy to take questions.
Aarno, thank you so much. What an extraordinary initiative and fantastic presentation. Really fascinating to hear not only sort of the mechanics of what you've established, but also the engagement with government and sort of the end-to-end initiation and engagement, across government, industry and academia to make FinnGen a reality. We've got a few questions already. I might kick off though. I'm going to take Chair's prerogative here. So with that government backing, I was particularly interested in that, and just even the idea of engaging across 3 ministries mildly makes my head spin, but regardless, we'll set that aside. I was interested that one of the first steps that you addressed was a legislative framework around FinnGen. And I was particularly interested in the secondary use of data legislation with the broad consent that data donors and participants have agreed to. Does the legislation provide protections in that, particularly in the context of a public-private partnership? So, for example, against genetic discrimination, use of information for risk-rated insurance, etc. How do you ensure the adequate protections are in place? Are they legislative?
Yes, they are legislative. There are clauses in both of the laws. I'm not a lawyer, so I don't remember them by heart now, but there are clauses that this may not be used for instance, for how do you call discrimination? Absolutely not, but also not for insurance purposes or these kind of things. Of course, Finland has a national healthcare system, so the insurances is not quite the same focus of discussion as it would be, for instance, in the United States. But there is legislative clauses against these. Yes.
Very interesting. I think that would certainly impact people's willingness to participate, having those protections in place. I guess the second part of my question along these lines, with that government buy-in and across ministries, do you believe, I note that you referred to, the use of polygenic scores and informing cancer screening or adaptation of the existing cancer screening policies and practices. Do you think having the buy-in of across these ministries is facilitating translation of your research and sort of health translation more broadly, health technology assessment in Finland?
I think so, that yes, there is kind of a backing. They especially the Ministry of Health and Welfare knows that something should be done. That said, I wouldn't brag that we have been really fast on those things, giving genetic data back, but we have initiated those type of studies that high risk breast cancer patients are called back and is first as a research setting, and the feedback has been really good. Actually, the one country that has practiced it even more and tested it even more is Estonia. They have had done a good job in testing that how does the public react to these type of recalls. And you need to engage, of course, the appropriate medical experts and community so that it's done the right way. The challenge is, of course, when you scale it up, that where are the resources to do it?
Yes, absolutely. Now, I could chat to you all day, but I can't monopolise you. So some other questions. So we have Amara working on Mexican genomics, wondering how other scientists can benefit from your biobank. Will it be publicly available, or will access be sort of fee for access?
Two ways. You can already now request access freely to the browser to the results, and you can download summary statistics freely. The only thing we like to have is that you register just for our bookkeeping, because we like to brag that 5,000 scientists already around the world have been downloading the data, and then when it comes to 10,000, we probably have a glass of champagne. So this is what we like to have. We like to distribute the data. Then it comes to the legislative challenges that the individual level data, unfortunately, we can't do or we don't have the permissions to do it like UK Biobank. But if there are specific projects of interest in both sides, so we have typically found the way to then collaborate with them so that the data would be more broadly accessible.
Okay, well, that goes to Sarah's question quite neatly. She's asking about the consent around for participation in the biobank. Can you speak to participants' opportunity to revoke consent, and with regards to data sharing, is it sort of the broad permanent consent, or do applications have an opportunity for it to be considered by participants before their data is shared? What degree of contact or dynamism do participants have with their consent?
That actually goes so currently that if you have consented to medical studies, there are a couple of things. First of all, you have the right to know to which studies your sample has been used. You can withdraw your sample and data at any point. Of course, if it has been given to some study already and they have done the analysis, that's kind of past, that's too late. But we always have a small number of individuals who withdraw them consent, 1 or 2 or 3 or 4 or in order of magnitude, but that happens.
Yes, okay. So Anonymous asks, what about the very rare variants that you can't impute accurately from arrays? These are typically the most clinically significant and highest effect size.
Correct. And those we can't identify unless they are one of those sitting on one of those Finnish enriched haplotypes. And so there's, in any study, you can't have everything. So this is the ultra rare variants and the de novos we miss.
No, fair enough. And that might also, there's another Anonymous asking about whether you hope to include proteomic analyses with your data sets.
Yes, actually, I didn't go into that, but that's exactly in FinnGen 3 that we plan to do proteomics specifically related now to these Finnish enriched coding variants. And we aim to look at the healthy blood donors where we think that the disease hasn't kind of affected too much the metabolism yet, so that we could potentially understand the early effects of these variants on biology. Little bit of metabolomics as well, but that's of less enthusiasm than proteomics at the moment.
You spoke in terms of the proportion sort of making up the data sets and the sources, about 3/5 come from hospital recruitment. Was that correct? 300,000?
Yes, roughly.
Okay.
roughly.
And hence the skew to people presenting with, you know, potentially genetic conditions. Yeah.
Correct, and especially with conditions that are related to age, because then again, these many of these what we call legacy collections, they have been collected the earliest in early 1990s. So so they are already quite old, many of them, and I think the mean age was 65 currently in the study.
And all adults, there's no pediatric recruitment?
No pediatric recruitment, no active pediatric recruitment, but we obviously have the health data.
Yes.
Also from the childhood.
Yes. All right, have to hurry up if I'm going to get through some of these. So is there a data retention time set for the genome data, or is it given the longitudinal data sets and ongoing research?
Superb question. So the question is, what happens after 2027? And this is something we are currently discussing with the ministries, and potentially even have to change the law a little bit. We can continue the research, but the question is that how do these big study sets, which have been originally developed for research, they become infrastructures? How can they be used as an infrastructure further on? And that needs work on the regulatory front.
All right, I'm conscious, Arno, that we're very close to time. I will leave the last word to my boss. All of our bosses, Catherine North, who is the lead of Australian Genomics, who simply says, amazing cohort, I'm jealous, smiley face. Arno, it's been just so wonderful hearing about FinnGen and having a chat to you today. It's an extraordinary initiative you've established, and I can see so many opportunities for ongoing engagement and collaboration. I definitely want to tee up a follow-up call and learn more about what you're doing at a more granular level and see what we can learn. So thank you so much.
Thank you, Tiffany. Thank you for everyone. And thank you, Will and Liana, for technical organization.
Thank you.
Have a great evening. Thanks.
Excellent. Bye. And thank you all for attending today's DNA Dialogue. Our next seminar will feature Crystal Tsotsi from Arizona State University, who is a Navajo human geneticist and bioethicist, who is going to be speaking to us about Indigenous data sovereignty. So that seminar will be on 30th of March at 9 a.m. Eastern. So I hope you'll be able to join us then. Thank you, everyone.